So what we're going to do here, looking at our urea cycle, is we are going to aim to basically get rid of nitrogen that originated from our alanine. Now, we took that nitrogen, we took that alanine, we did a transamination reaction, and transferred our nitrogen onto onto alpha-ketoglutarate to make glutamate. Now, nitrogen went onto glutamate. Then what we did was we used glutamate dehydrogenase in order to actually release that NH3. So that NH3, that ammonia, what we need to do is ultimately get rid of it. What that's going to do is we are going to use the enzyme known as ornithine. We already made the carbamoyl phosphate, but now what we're going to do is this step 2 right here is we're going to use the enzyme known as ornithine. Sorry, that's not an R. Ornithine Transcarbamylase. Now what that enzyme is going to do is it's going to use two different substrates. It's going to use ornithine and our carbamoyl phosphate. Now I'll ask that you look over at if you look at this ornithine molecule, one of the things that I'd like to draw your attention to is, well heck, it's got an N termini and a C termini. Well, this is an amino acid. Now, it's not one of those proteinogenic amino acids that we talk about so frequently, one of our 20 amino acids that we talk about, but this is nevertheless an amino acid. If you wanted to say what it most similarly resembles, well, you've got this long chain of CH2s with an NH3 on the end of it, so it's most similar to lysine, because lysine is going to have, rather than 3, it's going to have 4 CH2s there. Now, with that said, what I'd like you to think about is our enzyme, ornithine transcarbamylase. What it's going to do is going to take ornithine, which is able to be transported into the mitochondria, and it is going to take these groups right here, our carbonyl carbon and our NH2, and put it on the end of our side chain, of our R group, of our amino acid. Okay, so in order to do that, what we will release is our other inorganic phosphate molecule, because if you remember, we used 2 ATP in order to make carbamoyl phosphate. One of them resulted in the release of inorganic phosphate, and one of them, that phosphate group, was transferred on to make carbamoyl phosphate. So we're able to produce this molecule, citrulline. So that's ornithine transcarbamylase, which is going to do that. Now, citrulline is much like ornithine. It's able to be transported outside of the mitochondria fairly easily. Now, what citrulline is going to be citrulline is used for is, well, we're kind of going back to one of our products from our original transamination reactions. Remember, we deaminated, or we transaminated the nitrogen away from alanine, and we produced an alpha-keto acid. Pyruvate was that alpha-keto acid. What we're going to do with that pyruvate is we're going to use the enzyme pyruvate carboxylase to carboxylate that pyruvate and make oxaloacetate. Now, with that oxaloacetate, also known as OAA, we can use glutamate and do a transamination reaction and produce aspartic acid, or aspartate. Now, that aspartate can be transported out of the mitochondria through a transport protein known as the glutamate aspartate shuttle. And what we're going to do is transfer that aspartate, and we're going to basically add it to our citrulline. Now, what this aspartate is going to do is we're going to be replacing our carbonyl oxygen. Okay, so the carbon that originated from our carbamoyl phosphate, and I'm just going to highlight that so that you kind of recognize it's written in blue, but I want to highlight it just for the sake of visualizing it. We're able to transfer that aspartate onto that carbon right there. Okay, so that carbon, that you could kind of look at it as being a part of our side chain for our ornithine originally, because remember, this part of the molecule originated from ornithine. This part of the molecule originated from our carbamoyl phosphate. I'm just going to shorthand that as CP, and then this remainder of our molecule comes from our aspartate. I'm just going to write that as ASP. Now this molecule is known as arginosuccinate. Okay, now looking at that arginosuccinate molecule, one of the things that I'd like you to do is recall one of what we're trying to get rid of is our nitrogen from that alanine right off the bat. And our product is going to be the namesake of our cycle, and that is urea. Okay, so urea, pictured here, has 2 nitrogens on it. Well, with the addition of this aspartate to produce arginosuccinate, we've added our second nitrogen onto this molecule, or actually what truly would be our 4th nitrogen on here. Okay, so arginosuccinate is going to be kind of the precursor for our enzyme arginosuccinase. So arginosuccinase. What arginosuccinase is going to do is release fumarate. Okay, you might look at some of these molecules and be thinking, this is fairly familiar. I feel like I've seen this before, and you would absolutely be right. That fumarate, well, you'd be absolutely right. That fumarate, well, we've seen that before in the TCA cycle, or the tricarboxylic acid cycle. Well, this fumarate molecule is written in red, and that can go into the mitochondria and ultimately feed the citric acid cycle. Okay, so this hydrolysis event here done by arginosuccinase is going to produce the amino acid arginine. Now if we look at arginine, what does the R group of that look like? Well, we've got 3 CH2s, and sorry, we've got our N termini, our C termini, 3 CH2s, a nitrogen, and then this carbon with a double bonded NH2 and a single bonded NH2 with an overall positive charge. Well, what we're able to do with this at step number 5 is we're able to bring in the enzyme arginase. What arginase is going to do is it's going to use water in order to release this urea molecule. Okay, so when it comes to nitrogens that the urea cycle is releasing, well, urea cycle is taking 1 nitrogen from alanine and another nitrogen from an aspartate. Quite a bit of this takes place in the cytosol, but there are some steps that are involved, or that take place in the mitochondria. So the release of this ultimately is something that gets rid of this NH2, which ultimately could be, if not for this cycle, urea accumulation could ultimately be quite hazardous and quite potentially fatal. What this urea does is it goes to the bloodstream, and then it goes to the kidneys, where ultimately it is released in the urine. So humans are able to do this cycle and ultimately get rid of that otherwise potentially toxic compound, or those nitrogen-based compounds, that ammonia. All right, well, I hope this is helpful. This is the Urea cycle in a nutshell. This is something that plays into the metabolism of amino acids and the breakdown of amino acids. So basically what we've done with this, and looking at this, is we've looked at, well, in any given amino acid, but we look specifically at alanine, what happens to the nitrogen of that? Well, we release it in the form of urea. Now what we're going to talk about in a little while, or in our next video, is we're going to talk about, well, what happens to the other carbons and the other atoms in those compounds. Alright, well, thank you very much. Have a good one.